Hello students, welcome to Loksatta Yashasvi Bhava. In the last session, we have studied about the monohybrid crossing which was shown by Gregor Mendel. In this session, we shall study about dihybrid crossing and we shall also calculate the dihybrid ratio. In this case, Mendel performed the experiment by using garden peas, but he contrasted two pairs of characteristics. He used round and yellow seeds of peas and green and wrinkled seeds of peas. So there were two pairs of characteristics which were crossed. The P1 generation shows the formation of gametes. In the F1 generation, that is the first filial generation or the progeny, that is called as, we can see the formation of capital R, small r, capital Y, small y. Capital R indicates the formation of a dominant character, whereas capital Y is the yellow dominant character. Capital R is the round seed which is dominant, whereas small r is the recessive character. Small y is also the recessive character. Thus, there are four types of gametes which are formed. In this tabular representation, crossing of gametes can actually be studied. Students, remember, you only have to multiply each and every gamete with the gamete that is written on the top. Always write down the dominant character first. That is, we will begin with the R, capital R written first. If there is no capital letter or there is no dominant character, then it can be represented by a small letter which indicates formation of a recessive character. After studying this or after crossing this, we come to the phenotype which shows the physical characteristics or the characteristics that appear. In this case, capital R, capital Y. There are 9 capital R's and capital Y's if you calculate. If you count small r and capital Y, there are 3. Capital R, small y, there are 3 characters again, and with small r and small y, there is 1 character. So thus we have the phenotypic ratio as 9:3:3:1. And now the genotypic ratio, that is the genetic constitution. Here you can actually count the number of capital R's, capital Y's, small r's and small y's, or a capital R along with small y. And this is how it can be represented. Same thing can be seen in case of humans. Let us see how sex determination happens in case of humans. Chromosome is a thread-like structure that is present on the nucleus in the cell. Chromosome is formed of DNA, that is deoxyribonucleic acid. A fragment of chromosome is called as genes. How sex determination happens in humans? In case of humans, there are 46 chromosomes. That is, there are 44 autosomes, same type of chromosomes, and 1 pair of sex chromosome. Chromosome is a thread-like structure that is present in the nucleus of the cell. Genes are present on the chromosomes. Chromosomes are formed of DNA, that is deoxyribonucleic acid. A gene is a hereditary character that gets transmitted from parents to offsprings. There are billions of genes present on the chromosomes. In case of human beings, there is 1 pair of sex chromosome. If the X chromosome of a man pairs with the X chromosome of a woman, then it results in the formation of a female child. Whereas if the Y chromosome of a man pairs with the X chromosome of the female or a woman, then it results into the formation of a male child. Hence, sex of the child depends entirely on man. Students, after studying sex determination in humans, let us now study what is evolution. The changes taking place in the development in an organism over the period of years is called as evolution. There are various evidences that support theory of evolution: morphological, anatomical and paleontological. In case of morphological evidence, it states that even though there are millions of plant species which have got developed, all of them have the same or similar morphological structures like roots, stems or leaves. Thus, the morphological evidence supports the theory of evolution. In case of anatomical evidence, also the bones that are present in case of humans, in case of animals, in case of birds or fishes might be different in their shapes, but they perform this similar functions. Thus, the anatomical evidence also supports theory of evolution. Last but not the least, the paleontological evidence. Paleontology is the study of fossils. Fossils are the dead remains of plants and animals that get buried under the rocks. The impressions that get formed under the rocks proves that there is a series of changes and developments that has been taken place over the period of years in the life of living organisms. Thus, the study of fossils also supports the theory of evolution. Students, now we shall study about vestigial organs. In certain plants and animals, there are certain ill-developed structures or organs which are non-functional. These are called as vestigial organs. For example, in case of humans, appendix is an example of a vestigial organ. Now, we shall study about Charles Darwin and his theory of evolution. Charles Darwin was a British naturalist who performed a lot of experiments to study evolution. He studied a number of species and their development. He also stated the theory of survival of the fittest and also the theory of natural selection. In case of survival of fittest, he stated that the organisms, that is the plants and animals, will only survive if they are fit. That is, the fittest of the species will survive, whereas the others will die. And these fit species will then reproduce and pass on their characteristics to the next generation. According to the theory of natural selection, the criteria for selection is not deliberate in case of natural selection, and also the criteria is only for successful adaptation for growth and reproduction. The theory of natural selection can be explained with the example of black and grey moths that were found in England in Birmingham. There were black and grey moths which were found in Birmingham in England. These black and grey moths were found on the trunks of the trees. There were also a lot of algae and fungi on the trunks of the trees which were grey in color. Hence, the grey moths were camouflaged and the black moths became visible to the predators, and so the number of black moths consequently reduced, whereas the number of grey moths increased. After a period of time, it so happened that after the Industrial Revolution, pollution increased due to the industries. As the pollution increased, there was a lot of soot that was getting formed into the atmosphere. Soot is nothing but carbon dust which is black in color. Gradually, the trees started getting covered with black dust. Then it so happened that the black moths which were there on the trees were camouflaged, and the grey moths started becoming visible to the predators, and then the grey moths were eaten by the predators. Thus, the number of grey moths gradually reduced and the number of black moths increased. The example of black and grey moths explains the theory of natural selection. Students, we have studied about mapping of genes in these sessions. If you have any queries regarding this chapter, please send your queries to loksatta.com.